How do we learn? What makes the brain resilient after injury or trauma? What can we do to optimize learning? These are the questions that fascinate me and form the basis of our research in the Brain Plasticity Laboratory. I'm Dr. Sangeetha Madhavan. I'm a brain researcher in the Department of Physical Therapy. The brain is constantly learning and changing, a concept called neuroplasticity. The brain has formed new neural connections as you started listening to this talk. The science of brain research is also constantly evolving. Things that were true 10 years back are now not true. For example, in a healthy or neurotypical brain, the left side of the brain controls the right side of the body. Recent research, including work from our lab, now show that this is not the case in an adult brain with stroke. We now know that in some people, other areas of the brain can take over after injury. That is, the contralesional or unaffected hemisphere can control the affected side of the body. We use advanced brain imaging techniques such as functional magnetic resonance imaging and transcranial magnetic stimulation in our lab to understand neuroplasticity or brain reorganization after stroke and other brain injuries. We just received a new grant from the National Institutes of Health to explore pathways in the cortico-reticulospinal system that have never been studied before. I just told you how awesome neuroplasticity is. Then why do people not recover fully after brain injury? What limits and facilitates neuroplasticity? These are other questions I study, specifically about how it relates to walking recovery after stroke. Despite significant medical advances, current rehabilitation interventions for walking after stroke do not fully work. As a result of this, stroke is the leading cause of long-term disability in the world. So what can be done? The approach my research has taken is to boost neuroplasticity using external interventions to support learning. We can increase the activity or signaling between neurons, a phenomena we call cortical priming. For instance, when you smell the scent of vanilla and immediately get hungry for a cake or a cookie, this is an example of subliminal priming. Priming interventions we study include aerobic exercise, brain stimulation, and ischemic conditioning. My research has developed some of the foundational parameters of priming the brain to enhance walking recovery after stroke. We have been continually funded by the NIH and American Heart Association for the last 10 years to keep this work going. As a physical therapist by training, I like to ensure that these protocols can be easily adapted by clinicians with minimal cost and low risks to patients. But there are some barriers to what I do. Variability. Variability in how patients respond to these interventions. This is not surprising given how complicated the brain is. One single intervention is not going to work for everyone, which has led me to the path of personalized medicine. To optimize outcomes, each person requires their own type and dosage of intervention. My research shows that there are certain traits in brain structure and function, and not only that, but also genetic characteristics, which determines how patients respond to rehabilitation. Understanding these individual differences is the next direction we are heading towards. Understanding these individual patterns of learning is going to enable us to develop more successful interventions that are patient-centric for brain reorganization and walking recovery.